Hi everyone, my name is Rachel Culp-Hill and I'm a graduate student at the University of Colorado Anschutz Medical Campus. I study biochemistry and my research uses two things that we're going to be talking about today, metabolomics and mass spectrometry, to study how we can treat different types of cancer a little bit better. Metabolomics is a combination of two words. The first part comes from the word metabolite, which is a small molecule important for the survival of all the cells that make up your body. The second part, omics, means to study all of something. So together, metabolomics means to study all the metabolites in your body. As you might have already learned, when we eat, small molecules like glucose are broken down to make energy. One way glucose is broken down is by a pathway called the Krebs cycle, which turns the glucose into ATP, a molecule that has a lot of energy. But the Krebs cycle isn't the only important pathway for cells. As you can see here, each dot represents one metabolite, and they're all connected. Each of these metabolites is important for a cell to survive. When we're healthy, everything is in balance, but when we get sick, we can either have too much or too little of some of these metabolites. But these metabolites are tiny. If you imagine that you were as tall as the Earth, a glucose molecule would be the size of a marble. So how can we see them or tell that they're out of balance when we're sick? We can do this with a technology called mass spectrometry. Here are some real-life pictures of the mass spectrometer I use in the lab. First, we can take samples from people and put them into this tray. Then in number 2, they're injected into a whole bunch of tubes and pumps before going through what's called a column at number 3. The column helps slow down some of these metabolites so the computer at the other end doesn't get overwhelmed. Then at number 4, the sample gets to what's called the source, which uses electricity to get the metabolites into the mass spectrometer. Once they're in the machine, we can see the metabolites. On the left here are 3 different ones: ATP, caffeine, and glucose. The metabolites fly through the machine, and a powerful magnet repels them in a different direction. Now, metabolites are different sizes and shapes, so when they get repelled, some don't get pushed away by the magnet quite as much, and they spread out like a fan. Finally, those metabolites hit a detector, and a computer is then able to tell us how much of that metabolite was in our sample. With this information, we can compare 2 groups of people. For example, because we use energy when we exercise, our level of ATP is probably going to be lower than before exercising. Or, right when we get up in the morning, we probably won't have much caffeine in our body, but that's probably going to go up after drinking some coffee. And glucose levels are probably also going to go up a little bit after we eat some dessert. This information is really helpful to find out how to treat different diseases like cancer, diabetes, and more.